right, so we've reached kind of the end of our genetic line here. We talked about DNA, we talked about RNA, now we need to talk about proteins. We need to talk about the function of proteins and what they do in cells and different tissues. We need to talk about what amino acids are and how they relate to proteins. We're going to talk about what we call the 4 levels of protein organization. And this has to do a lot with protein shape. And as we said in chemistry, right, shape is everything. So the shape of a protein dictates how it's going to function. And we'll talk a little bit about an example where when proteins don't go into the right shape, negative consequences can occur. All right, so what do proteins do in the cell? Proteins are some of the most abundant biological molecules, and they have a lot of different functions. All proteins, though, are polymers of units called amino acids. You may have heard of amino acids before. We don't need to memorize all 20 amino acids that are here, but amino acids are the monomers that get stuck together to build a protein polymer. There's lots of different things that proteins do. Many proteins are enzymes. Remember, enzymes help catalyze reactions. They help them happen. So in you have a lot of digestive enzymes that help break down your food or things like that. They can be involved in transport, moving things throughout the body or across membranes, things like that. Structural roles, these things called actin and tubulin, they form like a little rail network inside of your cell called the cytoskeleton. There are proteins that are hormones. You've probably heard of this hormone, insulin. Insulin is important in regulating metabolism and blood sugar. If you are diabetic, you do not make insulin, so you can't regulate your blood sugar. You have to be provided with insulin. We'll talk about that in this course. There's defense of the body, things called immunoglobulins, like antibodies. These are proteins that help protect us from pathogens. There's some that help cells contract. Actin and myosin are key in muscles to help them contract. There are things that store nutrients like egg white, what we call the albumin, that stores nutrients for the developing embryo. So there's lots of different functions and there's many, many different proteins. I'm going to talk again about enzymes because they're very, very important, particularly when we talk about metabolism. All right, so remember, enzymes are protein catalysts in biological reactions. So all enzymes are catalysts, not all catalysts are enzymes, because proteins that are catalysts, we call those enzymes. They take in a substrate, and they have a region called the active site. The molecule binds to the active site, and then it kind of puts it in a proper shape by bending it, essentially. And then in this case, that allows these two pieces to be broken off from our substrate, and we call those our products. For this to occur, the enzyme has to have the proper shape. Here's another example of an enzyme that's very specific. This is endocellulase. It breaks apart cellulose. You don't make this enzyme, right? You have another one, amylase, that breaks down starch, but you don't have one to break down cellulose. So you cannot digest cellulose into glucose. Otherwise, we could go out and eat grass like a cow and survive. Although, remember, in cows, it was the bacteria that do that. There are 2 broad types of enzymes, and we're going to think about this in terms of, do they put things together or do they break things apart? If enzymes are catabolic, that means they break something apart. Here's an example of a catabolic enzyme. We take our substrate, it comes in, it gets broken apart. That's catabolism, the breakdown of substrates. The other type of enzyme do the opposite. They are called anabolic. Okay, they build more complex molecules. So if we took these two pieces and stuck them together, that would be anabolism. So enzymes are some of the most important proteins that we're going to talk about. There are other proteins that do all kinds of things, like I mentioned previously, but I want us to know this because it's important for metabolism. What are proteins made of? They're made of amino acids. Okay, just so you know, If I abbreviate them AA, I'm talking about amino acids, not talking about anything else that has the initials AA. Don't worry. There are 20 different amino acids. There's different ways that we talk about them. Here's a chart with their full names: glycine, alanine, valine, things like that. But there are different codes for them. There's a 3-letter code or a 1-letter code. So like valine is V in the 1-letter code or VAL in the 3-letter code. But we run out of letters because there's lots that start with A. So aspartic acid has a 1-letter code of D and a 3-letter code of ASP. You do not need to memorize these codes or their structures. That is for your evil 400 level biochemistry professor to make you do. Although I don't have them memorized, but it is good to know that there are different shapes of amino acid, and that gives them different properties. Like these 3 down here have these big rings on them. That means they're real large, and they kind of kink proteins in various ways. These cysteines can form little disulfide bridges because they have that sulfur there. Glycine's really small there. So just to note that they have different charges, different properties, and from the different groups here. So the basic structure of an amino acid is that it has this backbone here. It's got an amino group, that NH2 here. Then it's got a carbon and another carbon and this carboxyl group here. And then that's the backbone. Onto that we stick what's called the R group, or the side chain. That's each piece that's highlighted in blue here. That's what's different between the 20 different amino acids, and that gives them their different properties. For humans, there are some amino acids that we cannot make. There are 9 essential amino acids. You cannot make them. You have to eat them from other sources. So those are called essential amino acids. Don't ask me what they are off the top of my head. I can't remember. But there are other organisms, like certain bacteria, that can make all of the amino acids. So they're very important because they make those amino acids, and we have to eat them then to get those. When we stick together amino acids, we start building what we call a polypeptide. A polypeptide is a chain of amino acids joined together by a peptide linkage. So when we take one amino acid and stick it into another, we're building a polymer. So it's a dehydration synthesis reaction. We call that a peptide bond. It joins them together. And when we get many amino acids stuck together, we call it a polypeptide. Technically, a protein is one polypeptide or multiple polypeptides together. I'll come back to that in a moment. I'll show you some diagrams to explain that difference there. I might use these terms interchangeably. Technically, they're different. Don't worry too much about it, but know that a polypeptide is a chain of amino acids joined together with that peptide linkage. All right, so how they get stuck together is only a small part of the overall equation for what a protein does and how it functions. There's actually 4 levels of protein structure. You can think of this as 4 levels of protein shape. We're going to go through each of these. I would like you to know each of these here. All right, at the first level, the primary level, this has to do with the linear order of amino acids. I'll show you an example in a moment. The secondary level has to do with kind of localized hydrogen bonding and folding that causes these 2 structures, alpha helices or beta sheets. We'll come back to that one. The third level, the tertiary, this is an important one. This is overall the 3D structure that the protein folds into. This is determined by many factors, including what amino acids are in there, but also the conditions that the protein is in. Sometimes we stop here. Sometimes this is the last level, the third level. But you can go one higher level called the quaternary, or 4th level. This is when you have multiple polypeptide chains that interact with one another. So here you have the light blue interacting with the dark blue. Those are 2 separate polypeptide chains. Okay, let's look at this in more detail. So let's start with the primary sequence. This is the order of amino acids. We'll learn all about how this is determined. Most polypeptides are somewhere in the range of 100 amino acids up to thousands of amino acids long. So this is real short here, but it's just a diagram. We have the N terminus, the amino terminus. That's where it starts, and then it runs down the chain to what we call the C terminus, the carboxy terminus. That's the end of it. So it goes N to C in order. And then you can see here, each of these is an amino acid. So here's methionine, here's aspartate, here is isoleucine, here's proline, glutamine, threonine, lysine, glutamine, right? So on and so forth down the line. Okay, so that's the linear order of the amino acids. If I were to have a methionine and then instead of aspartate, another methionine, that would change the linear order, right? Why does this matter? Well, there are some proteins that change very little over time. And that can be really cool when we think evolutionarily. Remember I talked about that term called phylogenetics, where we're trying to use DNA sequence and things like that to understand how things are evolutionarily related? Well, here's a perfect example of that. We're not using DNA here, we're using the protein, but remember DNA codes for the protein, so it's the same thing basically. And this is a gene called cytochrome C. It's part of our cellular respiration. It's what allows us to make ATP and to keep living. If you break this gene, cytochrome C, you generally die. So over evolutionary time, it does not change a lot. Here, we have the primary sequence of our amino acid for cytochrome C for the polypeptide. You can see humans and chimpanzees, our sequence is identical. So we're on the same line here. Us versus rhesus monkeys, very, very similar. Here, I spot a difference right here. We have a threonine versus an alanine there. But other than that, it looks identical there. So over the however many millions of years between our evolution between rhesus monkeys and humans, there's only been one amino acid that changed there. Now, if we go down even further, we go down to something like a plant. You can see that, yeah, the sequence has changed, but actually not that much. That's kind of crazy, right? Like, that could be quite a long time, almost 1 billion years between our last common ancestor, and we still are very, very similar in terms of sequence there. So looking at these sequences can help tell us how long ago these changes might have happened and how related we are to other organisms. This is part of a field called phylogenetics, where they are basically doing those kinds of things and looking at these changes to see how closely related things are. All right, let's talk about an example that might interest you and you probably have heard of. This is an example where changing one of those amino acids can cause huge downstream effects. So in our body, we have red blood cells. You're probably aware red blood cells carry oxygen, right? I think we all know that one. I even know that one. I don't know a lot about human anatomy, right? In the red blood cells is a protein called hemoglobin. And hemoglobin is this, it's actually several chains, but in it, it has its single primary sequence, right? The order of amino acids. Normally, there is a region that goes threonine, proline, glutamic acid, lysine. That's the order. When that happens, everything is fine. Your red blood cells make hemoglobin just fine. If there is a mutation that changes this sequence, and instead it goes threonine, proline, valine, lysine, see how this is different shaped? This one's pretty big, this one's smaller here. That leads to downstream changes in the protein itself. This change here, this amino acid substitution, causes a disease called sickle cell anemia. That disease is because the hemoglobin protein forms these long chains that lead to the red blood cells, and instead of being this nice donut shape, it leads them to be these real long, skinny, sickle-shaped cells here. those cells can block up blood vessels and cause basically clots to happen, and that can be deadly. So this disease is caused by a mutation. Now, very, very interestingly, you may have heard that recently there have been some trials of a gene therapy where we use a technology called CRISPR-Cas9 to come in and change the DNA sequence of people that have sickle cell anemia. We change this mutation back and hopefully fix the problem. And there's been a few people that have been cured of this very, very recently. So promising new technology here. Although it brings up all kinds of interesting ethical questions about should we be changing people's DNA and things like that. I think we probably can agree that in the case of disease, yes, we don't want people to die of sickle cell anemia, right? But then, what are the other consequences of being able to change people's genome? We'll talk more about that when we understand a little bit more about genetics later in the course, but I just want to plant that in your brain there that this is a change in protein sequence that causes big effects. All right, our second level of protein structure, the secondary level, is local folding. So small little regions can fold up into shapes. Sometimes they will fold into these helical structures we call alpha helices in regions. They'll coil up in these helices. Other times they will be these flat beta pleated sheets here. These are caused by hydrogen bondings between the amino acids in there. So this one we're not going to go too much into detail, but it is good to know because you will see these coiled structures and these flat structures in there. That's the secondary level. When we look at the polypeptide as a whole, here's where we're looking at the tertiary structure, which is the overall 3D shape of the protein. We just got done talking about our hemoglobin, right? Here is properly folded hemoglobin. You see it's kind of circular here, and it's got this nice big space in here. Here's improperly folded hemoglobin. It's kind of all stringy and a mess, right? It's kind of collapsed all over itself. The folding that happens here in the third level is due to the interactions of those R groups. So here you can see these two R groups are forming an ionic bond together, causing this big loop here. Here we have two that are caused by hydrophobic interactions. This one, two sulfurs cause a disulfide bond here. Fun one about this, these disulfide bonds, if you have curly hair, those disulfide bonds are actually causing the curls in your hair down at the protein level. And if you straighten your hair using a straightening iron, you are breaking these disulfide bonds by applying heat to your hair. So that's a fun one there. When we have a properly folded protein, it does its function. Unfolded proteins are called denatured. A lot of proteins that become denatured cannot refold properly. Some can, others can't. Depends on the protein, the conditions, but if they're not in the proper shape, they're not going to do their proper function. So I used this example previously. There are many different things that can cause denaturation of proteins. We use the example of the egg white, right? Egg white has protein in it, the albumin. When you apply heat to it, that denatures the proteins, causing them to solidify. They do not refold, right? You can't go back to having a liquid egg once you've cooked it. Some other things that can cause proteins to denature are extreme pHs, basic and acidic, salty conditions, and certain chemical conditions. That folding, right? Remember we talked about that folding causes the shape. Here's my favorite protein again, green fluorescent protein. It folds into this barrel-like shape, and in the middle here are some amino acids and some atoms that when they get hit with certain wavelengths of light will emit a green glowing color. This is called fluorescence. This protein can be added, the DNA sequence can be added to pretty much any organism. Here's a bunny with GFP added to it. Here's a fish This is probably maybe a mold or something. Here's some cells that have it added. These look like human skin cells or something. Here's a plant with GFP. Here's a worm, a fly, some little naked mole rats or something like that, right? So this can be added to anything. That's because we all share the same code. The code is universal, and we're going to learn about how to decode things and how to read DNA and stuff like that. But our DNA can be read by any other organism because we all share common ancestry, right? We've all evolved from the same system, so our systems all work the same. That's really, really cool, right? Taking this protein from a jellyfish originally, and you can put it into a plant, and it still works. That's crazy. That's super cool, right? So this is our third level, that 3D folding. We do have a fourth level, the quaternary structure. This is when we get multiple polypeptide chains interacting. This doesn't always occur, but in these things called antibodies, antibodies have 4 different polypeptide chains. They have 1, 2, 3, 4, and they interact with one another. The cartoon diagram, they look like a Y, but they kind of look a little bit crazier here in the actual 3D diagram. These 4 chains interact with one another, and that's kind of cool because you can sometimes differ the forms by swapping bits out and mixing and matching and stuff like that. So they can have slightly different functions then. Okay. Please make sure you know these 4 levels of protein structure. I will ask you about them on a test for sure. All right, one little bit here to finish up with. Protein folding is really important. I just got through telling you all about it, right? Protein folding is reliant on the order of amino acids, but also the place where the folding is happening. Like I said, if it's too hot or too salty or things like that, a protein might not fold properly. So in your cells, there are special little proteins called chaperones that can help proteins fold. So a protein might get made by a ribosome, and then it goes into this little barrel-like structure called a chaperone that provides a nice, safe environment for the protein to fold properly, and then out it pops, all ready to go and do its function. So there's a lot of importance put on proper protein folding. Okay, let's test ourselves here. So enzymes that do dehydration synthesis reactions are which of these things? Symbolic, anabolic, catabolic, hyperbolic. Go ahead and pause the video. Think about this. All right. Dehydration synthesis, we're putting things together. So that would be anabolism. Anabolism is building larger molecules by putting monomers together. So an enzyme that does dehydration synthesis reactions would be anabolic. All right. So proteins, they're polymers of amino acids. They're also known as polypeptides. They are formed when one amino acid gets peptide bonded to another one, and we keep building that chain. There's 20 different possible amino acids that can be in there. They vary in what we call their R groups, which give them different properties. They can have a diverse range of functions. Many of them are enzymes, which are our biological catalysts. Protein folding is very important. We laid out the 4 levels of protein folding structure. Primary, that's the linear order of amino acids. Secondary is local folding into alpha helices or beta-pleated sheets. The tertiary level is the overall 3D structure. That's determined a lot by the interactions of the R groups. And then our quaternary structure is when we have multiple polypeptides that interact with one another. Denaturation is unfolding or improper folding of a protein, and we said that there are special proteins called chaperones that provide favorable environments to make sure that proteins fold properly so they can do their function. All right, that's the end of chapter 3. We made it through our overview of our biological macromolecules. Make sure you know those really well because we're going to come back to each of those things because they're critical to how cells function, right? They're what make up the cells. We've got to understand the building blocks to understand the larger picture of the cell here. All right, that's it for chapter 3.